Thanks. I'm Michael Stein, CEO of Valo Therapeutics. I'm going to be really talking to you about the next generation, if you like, of oncolytic viruses as immunotherapy. Essentially, what we're doing is developing a rapidly adaptable platform to treat solid cancers. We're using two platforms which are very similar, and I'll come to those in a moment, called Peptic Ret, which stands for peptide-coated conditionally replicated adenovirus, Peptic Ret, and Pepti-Env, which is a very similar concept where we're coating envelope viruses with peptides that are, of course, tumor-specific. So that's the basis of the technology, that both platforms have been patented. The technology has been actually licensed and now acquired from the University of Helsinki out of the laboratory of Professor Vincenzo Cerullo, the Professor of Immunology in Helsinki. So far, we've raised $9.5 million, half of which has been non-dilutive from the Finnish government, match funding the equity of approximately 4.6 million euros. And we're now seeking our Series A, starting in Q1 of this coming year, to take us through the clinic for a phase 1 trial. So the team, which I lead out of Oxford, my CBO, Scott Cuthill, very experienced CBO in the pharmaceutical industry, is also based in Oxford. So the commercial team's Oxford-based, and our highly experienced R&D team, led by Sari Pessinen, who's the whole team very experienced in the oncolytic virus space and in the immunology space, is based in Helsinki. Finland's an unusual place in terms of biotech. It certainly has a very specific niche in oncolytic viruses. There are approximately 3 oncolytic virus companies that have come out of Finland in the last few years. So there's a lot of talent pool with that specific idea in mind. So the basic technology is the following, that on the left-hand panel, you've got the peptides, which we use a polylysine tail, which are net positively charged. These are lysine residues, which we attach and tag the peptides, and those are the tumor-specific peptides. And we then combine them with the negatively charged capsid of an adenovirus. And that's the technology, which is a charge interaction. There's all sorts of technology around that in terms of the relationship of the specific amino acid residues in the peptides in relationship to the number of lysines you have to add, as well as to the constituents within the buffer that we've worked out over the last 2 years. And you can then put any selection of peptides with a polylysine tail onto a stock virus, adenovirus. And the complexes, which you'll see here on your right, are the Peptic Ret complexes, which are stable through freezing and thawing. And it takes about 15 minutes between combining the virus in its buffer together with the peptides to create those stable complexes. So there's a clear advantage, if you like, over other competing therapies such as CAR T and other more sophisticated technologies that allow us to do that process very quickly, given that it only takes about 2 to 3 weeks to GMP manufacture peptides. We have a very similar concept in relationship to Pepti-Env, standing for, remember, peptide-coated envelope viruses. So this is now applying it to the envelope viruses using a different tag, a cell-penetrating peptide or anchor. But it's exactly the same kind of concept applied again to envelope viruses. The advantage there is that envelope viruses tend to be much larger, allows you to put on many more different types of peptides onto a single virus. Within Peptic Ret, the adenovirus, we can put up to approximately 5,000 peptides, individual peptides per viral particle, so you can deliver many millions of specific peptides into the tumor through intratumoral injection. With Pepti-Env, you can times that by between 5 and 10-fold in terms of the numbers. So that's both technology platforms are patented. And of course, if you think about it in terms of the tumor specificity, it's in the peptide, which is the bit that you can manufacture, as I said, within 2 to 3 weeks. So we really positioned uniquely as the next generation oncolytic virus company. So we really regard ourselves as an immunotherapy company, not the oncolytic virus company, because we're agnostic as to the virus itself and open to partnering with different companies in the oncolytic virus space who are interested in immunotherapy by tagging the oncolytic virus with the peptide. And in the era of checkpoint inhibitors, which I'm sure you're all aware of, the checkpoint inhibitor essentially is taking the brake off the immune system, but non-specifically. And our technology platforms allow you to then direct the immune response and enhance in advance the CD8 positive, the killer T cells, specifically targeting the cancer. So that's our real niche, and I'll show you some data that we've generated. We're preclinical stage, but we've got some very encouraging data that I'll show you. So essentially what we talk about is a versatile plug-and-play immunotherapeutic platform to trigger robust tumor-targeted immune responses to any tumor antigen. And that is the hypothesis that would be much more effective than existing oncolytic platforms. So we work very synergistic with checkpoint inhibitors. There are a number of factors which I won't really dwell on. I think this is a pretty sophisticated audience, but essentially just to point out that we can take checkpoint inhibitor naive or refractory patients and using our approach actually turn them into responsive patients. Because we actually, most people fail checkpoint inhibitors either because of side effects, so you want to be able to reduce the dose, or because you simply don't have the tumor-targeting immune cells available once the checkpoint inhibitor has taken down the barrier to immune detection of the cancer. So the idea is that the checkpoint inhibitor takes the brake off the immune system. You've now got the tumor-specific T cells present, and you should then get much more efficacy across patients that are refractory or naive in terms of checkpoint inhibitors. The other point, of course, from a commercial perspective, in terms of, for example, cost of goods, this approach means we can take a standard stock oncolytic virus and attach any peptide to it within 15 minutes. That's the process. So 2 to 3 weeks to GMP manufacture the peptide. Of course, it takes a lot longer to GMP manufacture a virus, a live genetically modified virus. And so this technology allows us to have the stock virus already ready to go with your checkpoint inhibitor. So in future, we'd be able to not just use tumor-associated antigens, which might be present on a number of different kinds of solid cancers, but you can even then go into think about the personalized medicine space where you can then deep sequence the individual's cancer, which is happening more and more, of course. And you can then, instead of waiting 2, 3, 4 months for your CAR T and the potential toxicity related to that, you can, in the interim, use Peptic Ret or Peptic to actually deliver the same patient-specific modality if you know the specific neoantigen you want to attach to the virus. So really it's a very potent, potentially potent delivery platform for the personalized medicine space going forward. Our first stage, though, is to, I think everyone's aware of the checkpoint therapy market size. I won't dwell on that. It's obviously going from 14 billion to about 34 billion by 2024. There's a lot of activity around the oncolytic virus space, some recent deals at the top in 2019, a lot of excitement in terms of the combination of the checkpoint inhibitor with existing oncolytic viruses. And again, reiterating what makes us unique is the immune targeting, the tumor targeting aspect of our platform. So the pipeline at the present time, we're preclinical stage. We've partnered with Dnatrix, a company based in San Diego, which is an oncolytic virus company. Deep experience of space. We're using their virus, which has already been in the clinic, in our first program targeting melanoma. And then we have our own program of a different virus, which I'll come to in a moment, that is doing the same thing, targeting potentially different indications. And then we've got earlier stage, the Peptic platform, which is the envelope virus platform. And there we're specifically looking for partners. We believe that from an oncolytic potential perspective and as an adjuvant, because we believe that in the context of Valo Therapeutics, we're using the virus as an immune adjuvant for vaccination. And really then it's a vaccination strategy. And we believe a prime boost strategy would be very effective using envelope virus in combination with adenovirus. Adenoviruses are more potent, if you like, as oncolytic viruses in themselves versus typically envelope viruses. There's lots of activity in the space. But we are, as I say, agnostic to the virus and really trying to focus on Peptic Ret and Peptic. This is our own virus, which is the Valo D102 armed platform. And you'll notice here that we've got 2 immunostimulatory inserts, OX40 and CD40, again, because that's going to our strategy of tumor-targeted immune stimulation, where the OX40 ligand, we know, enhances CD8 positive T cell responses, and the CD40 ligand stimulates the antigen presentation, the T helper arm of that function. So The rest of it is typical to most oncolytic adenoviruses. But we think that one is particularly suited to the Peptic Ret approach. The mechanism of action then, just to reiterate, is number one, the Peptic Ret or peptide tailored for each patient or tumor indication. Injected intratumorally, we believe that's superior to intravenous because, of course, you've got lots of circulating antibodies to these viruses that mop up very quickly. A lot of people trying to go intravascular route. We believe that you get a better vaccination through intratumoral route, and we have some data for that. The APC then takes up the complex, and then you get pre-selected tumor epitopes and expansion of the CD8 positive T cells, versus the oncolytic virus mechanism of action, which is the more standard one, which other oncolytic viruses produce without Peptic Ret. So what makes Peptic Ret unique is the peptide vaccine mechanism of action. So a little bit of data. This is in a mouse model. We're not yet in human. This is all preclinical mouse models using a B16 over mouse melanoma model. On the left-hand side, the y-axis is your tumor volume. We then give 3 prime injections and 1 boost on days 9, 11, 13, and 15. It's a very aggressive model, so the animals have to be sacrificed by day 33 in this particular experiment. And you'll see the mock, what you'd expect, of course, tumor goes out of control. You get some control with the virus, that's the typical oncolytic virus activity. And then if you get the Peptic Ret, you get much more efficient control. And that just shows the individual animal data for that same experiment. In terms of mechanism of action, if you focus your attention on the what's called TPR2 GP100, those are the tumor-specific peptides in this mouse melanoma model. And here we've got mock showing their positive controls versus the negative control versus the anti-tumor, which there's a very noisy background. We haven't reset the baseline. There's a lot of noise in biological systems. So here we've just given you the data as it is, with each bar representing an animal. And you'll see then the naked oncolytic virus in the middle. Again, no statistically significant response in terms of CD8 positive T cells versus with the coated antigen with TRP2 and GP100 coated virus, you see a very dramatic increase in the number of tumor-specific and antigen-specific CD8 positive T cells. And then the actual animal data showing the combination with checkpoint inhibitors. Why we're very excited about this technology is that you then can see that with the mock, it's again tumor volumes on the left. This one's going out to 29 days. Again, similar aggressive tumor, B16F10 mouse model. We're injecting intratumorally the Peptic Ret at day 8, and then again a few days apart, but this time in combination with the checkpoint inhibitor, which is a mouse anti-PD-1. And you'll see the open circles are the controls. The blue solid circles are with the checkpoint inhibitor. We see a little bit of control of tumor growth. The Peptic Ret is the orange circles, again, similar to the checkpoint inhibitor. But what's really quite dramatic is when you combine those, which makes perfect sense from a mechanism of action perspective, where you get dramatic control of the tumor. So with that in mind, we're really, the summary is that we believe this is the next generation oncolytic virus company. We have a novel, rapidly adaptable immunotherapy platform, both for adenoviruses but also for envelope viruses, Peptic Ret, Peptic Inv. They're both patented, and we've shown very clearly they're highly synergistic with checkpoint inhibitors. We're going out to raise our Series A, as I said, in Q1, but very interested to talk to potential investors at an early stage and get early expressions of interest. Thank you very much.